All right, thank you so much, guys, for coming. So today we are on lecture 3. We're going to do cells, organelles, and transport. When I looked at your LearnSmart modules, I think we'll focus a little bit more on the transport. You can have a few questions on the transport wrong, and a few organelles, okay, with the specialized organelles like microvilli. So we'll kind of try to go through that a little bit more detailed. All right, very good. So we all know the 4 parts of a cell, right? The 4 major components, starting with the cell membrane, right? Okay, this is the boundary of the cell, and of course it is selectively permeable, which means it will let certain substances in and certain substances out. All right, and the nucleus, of course, is where all the genetic material is kept. All right, all the genetic material is in there, and then the organelles that have specialized functions. All right, these are membrane-bound organelles. Some of them are not membrane-bound, like the ribosomes and the centrioles. Okay, but most are membrane-bound, and you all know most of these organelles. What are some of the organelles in the cell? What are the examples? What's that? Rough ER, which is rough emergency room? Yeah, you see what I'm saying? So one of the other thing, I'm glad you say that, because in the exam, you have to always spell these out first. Don't just always say RER or SER or whatever. You always have to spell it out first, then put the abbreviation in parentheses. Then you can use it for free after that. But okay, so that's one of the things to watch. And then what else? The mitochondria and all those things. So we'll talk about each of those today and talk about their functions. All right, and then of course the cytoplasm. Some of the questions that you had wrong had to do with the cytoplasm. What are the 2 parts of the, or the 2 major parts of the cytoplasm? There's the first part where everything is suspended. What's that? What's that from the modules? Where everything is suspended, because it's like a cell is like a balloon, right? With what in it? Yeah, fluid, which is called, starts with a C again. What's that fluid called? Where cytosol, yeah, okay. Because you kind of had problems with that when it was talking about the cytoplasm and the 2 components. And then whatever, the second main part of the cytoplasm is the cytoskeleton, which is made of microtubules, made of the protein called tubule. And these microtubules kind of hold and suspend the organelles in place, or when vesicles move, those microtubules are like, straight microtubes, like little poles that are, sort of scaffold things around. All right, so those are the 2 main components of the cytosol. Is the fluid, and then the cytoskeleton. Otherwise, if you had nothing to hold those organelles in place, they would all kind of sort of settle, and it would be just everything squished like that. Okay, so the cytoplasm is important in that, and it's an aqueous environment. So of course we're going to do characteristics of each of those 4 things, and the nucleus is a specialized organelle. That's why we mention it singularly together with the cellular organelles. Okay, so we'll go through each of those and talk about the function. Again, this is a hypothetical cell because it's got all the possible organelles a cell can have. There's no cell that has all these organelles in there, except the ones inside, which are, even the ones inside, some of the cells don't have them, and so forth. Okay, so this, which is another area where you kind of had challenges a bit in the LearnSmart modules. What are these little ones? Microvilli, okay. And then these big fat ones are? Microvilli can be confused for cilia, because cilia and microvilli are almost the same, but they have very different functions. So, but the big fat ones, like on a sperm cell, are called flagella. Okay, so in the LearnSmart modules, you guys had problems with what's that? Oh yeah, of course, yeah. Okay, cilia, but yeah, okay. It looks so long that maybe it's because the picture is so blown up that you know, flagella is what I immediately thought of. But yeah, okay, cilia. Okay, in the LensSmart modules we had problems, so sorry about that. I just saw the huge thing and I just thought flagella already. All right, okay, but yeah, so the cilia is, yeah, it is a little bigger than the microvilli, but okay. What's the difference between cilia and in terms of function and microvilli? Microvilli increases the surface area. All right, and cilia, what does cilia do? On top of the cell membrane, yes, such as in the respiratory system. So you would find this in the respiratory system, and then this microvilli, you'd find it in the which system would you find microvilli? Digestive. That's where you need surface area for absorption and you know, digestion. Okay, so respiratory, like all the mucus with the trapped particles in the respiratory system, is moved by cilia, and okay, all the surface area is increased by microvilli. So one of the things we can put there, of course, the flagella, which is what I went to. So it's missing one other type of organelle, which is the flagella. I think one of the cells I'll show later has one. All right, that's why I jumped onto it on the cilia. Okay, so those are all the possible organelles. So we'll talk about each of them, and this is just a 3D cell, you know, in there. All right, so functions of the cell is to you know, take care of all the reactions in the cell, like the metabolism, conversion of certain nutrients, synthesis of certain nutrients, and cells become specialized based on the proteins they produce. All right, so from chemicals within the cell, and also release energy, all right, in the form of heat that helps to maintain the 37 degrees C of body temperature, right? So all these reactions, especially reactions that break down energy, they do not use all the energy that's released by ATP, and this energy is used to heat up the cell. All right, synthesis of molecules, all right, they like certain proteins, all right, or make certain enzymes, which in turn affects the function of the cell, because depending on the enzymes or the proteins that are made, then that determines the function of the cell. All right, and we'll go through some of all those functions based on the organelles that do that function. Provide a means of communication. All right. Last time I was talking about the glycoproteins and glycolipids, which are, you know, proteins and lipids that are attached to carbohydrates on the cell surface that act as cell markers. All right. And those cell markers help the cells to communicate with like cells, all right, and also sometimes instruct the internal functions inside the cell. And we'll talk about a few types of, you know, this kind of cellular communication with different types of molecules, like cadherins and integrins. And we'll talk about what each does. All right, and then of course reproduction and inheritance. You can see all these are based on the characteristics of life. All the functions of the cell are based on those characteristics. They satisfy those functions we talked about, we listed at the beginning as the characteristics of life. So mitosis, what does it do? What is the function of mitosis? Yeah, it pretty much increases cell number, right? By replicating the cell to form identical cells, like cloning. What about meiosis? What is it useful for? What's the major function? Yeah, in terms of producing what? What do you need for reproduction? Yes, and where is that DNA contained? Yeah, and the nucleus is contained in a certain type of cell. What's that cell called? Gametes. Yes, exactly. I had to pull that one out of you. Okay, so the gametes, production of gametes, meiosis is responsible for that, because you get a haploid cell. which means it has half the genetic material. When you have a union of the female and male gametes, then you get a diploid cell, and you get a whole organism. So meiosis is important for formation of gametes, and mitosis is just important for cloning cells and replicating cells, such as skin cells, for instance, right? Skin cells, they slough off every 45 days, so you gotta make new skin cells, and mitosis is responsible for that. All right? So the plasma membrane is important. We talk about it first because this is what regulates what enters the cell and what exits the cell. All right? So of course it's going to enclose the cell. It's going to support all the cell contents. All right? Support all the cell contents, and it's selective barrier. All right? It will let certain substances in and let certain substances out. All right? So it's a selectively permeable. All right? And then of course plays a role in communication based on the glycoproteins and glycolipids. All right? So there is another function of the carbohydrates. All right? And we'll talk about two of them. All right? So of course it's a boundary between extracellular material, which is the extracellular fluid. All right? It's also an aqueous environment. All right? And then also demarcates the intercellular. Intra is inside the cell. And intercellular is the extracellular material. You see that? With an ER, inter is between the two cells. Intra is inside. All right? So the inside environment is also aqueous. The outside environment is also aqueous, the fluid that bathes the cells. All right? And then this is very important in the orientation of the phospholipids. All right? All right? And we'll talk a little bit about that because you kind of had problems in determining the hydrophobic and the hydrophilic part and which direction that faces and why that is. All right? So we'll talk a little bit more about that. All right? So the structure of the cell membrane is the fluid mosaic model. All right? Because it's got the very fluid 2 layers of lipids like this, and then it's got proteins in it. Sometimes some of the proteins don't go through. All right? So you got this kind of arrangement right here. So you got that. This is also it could be a channel protein as well, or it could just be a receptor protein. All right, that's attached to other proteins inside of the cell, and that will do certain functions. Okay, so when we get to it, we'll talk specifically about the carrier proteins and the channel proteins, because you kind of had problems with that as well, because there's a slight difference between the two. So it's a double lipid bilayer. So which is the hydrophilic part? What's that? The head. Okay, and the hydrophobic part? The tails. All right. All right, and those are fatty acids. That's why they're hydrophilic. And so why are the heads oriented out here? We talked a little bit about it last time. Yeah, that's the intercellular and intracellular fluid we're talking about. It's all aqueous environment, right? So you've got water here, you got water there. So that's why they're inverted like that, all right? And then because of these proteins embedded inside of it, also between the hydrophobic tail, it's cholesterol wedged in between there to kind of stabilize the membrane. What is cholesterol in terms of the kind of organic compound it is? It's a lipid, right? So it's wedged in between with the hydrophobic tails, and that's what gives it that fluid mosaic model kind of structure, because it's a very fluid arrangement with all the proteins that are embedded in and out of it. All right? So, and then cholesterol actually makes 20% of that. And it kind of gives just strength and stability to the fluid mosaic model. All right. And then, of course, we already talked about this. One of the things we had problems with, the hydrophobic, which are the non-polar tails facing inside, because both the outside and the inside is an aqueous environment. And then, of course, the hydrophilic tail, the hydrophilic heads faced outside. They got a phosphate, and the phosphate is slightly polar, it's charged. So that's why you have, that's why it's hydrophilic, because water is a polar compound. One of the things that water can do, because it's very small, it can cross the, the lipid bilayer, but not very fast, but it can still do that. Even though it's a polar compound, but because of the size, it is able to cross it. But then we'll talk a little bit later about special proteins called aquaporins that actually move water fast through the lipid membrane. All right. So cholesterol gives it flexibility. All right. It's wedged in there. And the do you remember the structure of cholesterol? What does it look like? Which other compounds does it look like? Or what is it the mother compound for? We said it's a lipid, right? What does it look like? 4 hydrocarbon rings. Remember the 4 hydrocarbon rings? That's why it's different from the tails, because the tails are, long chains, and so if you wedge a compound that looks like this, you see, like that, and then it's got this. All right, so that's cholesterol, and it's got a very long chain over here. Okay, so if you put that in there, then that's what gives it that fluid kind of model, because the tails are just like that, and then there's a hydrophilic head. All right, so you can see what I was talking about. These are the glycoproteins, glycolipids, and these, say, are carbohydrate chains, and they are used for cell markers or cell recognition. All right, and then you got a protein channel, and then here you've just got a transmembrane protein. Okay, sometimes it could be a carrier protein or a channel protein. So we'll talk a little bit more about it. The carrier protein will actually attach and move and change shape and move substances in. Okay, that's a carrier protein. It will attach to the substance that's moved to the molecules and change shape, and the once it changes shape, that substance is internalized. But a channel protein has got a certain size pore in the middle that will let only certain size molecule in there. All right, and it will transport it without actually attaching to it, like a carrier protein does. All right, I'll cover it a little bit more because there are challenges with it. The mediated transport, when anything is assisting with the movement. Okay, so those two differences between a channel protein and just a carrier protein, we'll talk about it a little bit more. All right, so that's the fluid mosaic model of the membrane. All right, so very good. So functions of the membrane proteins. All right, so we got protein molecules, all right, that are floating, and some of them are only on the outsides. Most of them are transmembrane, which means they go across the lipid bilayer. All right, especially the channel proteins and some of the G proteins that are enzymes that will affect the rate of reaction on the inside, and those proteins will attach to something that is not hydrophilic, that's not hydrophobic. All right, so that it will not be able to cross the membrane, so it will attach to a protein on the outside and affect the inside, what's happening. All right, so what are the marker molecules that we just talked about? What kind of compounds are marker molecules? Glycoproteins and glycolipids. Okay, And these are also attachment proteins, cadherins and integrins. All right, so I'll take a different color. Cadherins, does anybody know what cadherins do? What do they attach from the module? It attaches a cell to other cells, yes. That's very good. And then integrins, they attach cells to what? Yep, to other molecules. Yeah, they attach cells to other molecules. All right. And integrins are almost like, receptors are sort of specialized integrins, if you will, because a receptor, what will a receptor do? It will attach to anything that is not hydrophobic, something that is not able to cross through the membrane, and then it will affect the inside of the cell, the inside functions. So integrins are also used as communication molecules, because when those proteins attach to a certain molecule on the outside, that molecule has the ability to affect what's happening inside the cell. All right? So it's kind of like similar concept to receptor proteins, because receptor proteins will have a specific site, though, which is like an enzyme lock and key that will attach to that specific ligand. All right, so these will have a ligand attachment site, whereas the just generally integrins attach to any molecule, to a variety of molecules. All right, which doesn't have specificity like receptor proteins. All right, so receptor proteins are specialized integrins. Do you see that? Receptor proteins are specialized integrins because they will only attach to a specific ligand of a specific shape, all right, and then they will affect cellular mechanisms inside of the cell. And all these are usually transmembrane, which means they will span the width of the hydrophobic bilayer, lipid bilayer. All right, and then a bunch of them are enzymes. All these proteins that will cross, such as the ones that are attached to G proteins. They will activate certain enzymes inside of the cell, and then second messengers, which we will cover when we do the endocrine system. All right. So all these are the functions of the proteins that are waged within the, and we'll cover some of those as we talk about the rest of the functions, like mediated transport and so forth. Those are also proteins. All right. We'll define each of these movements of the cell. All right. So diffusion, what kind of movement is diffusion? What's that? Yep, it moves solid from higher concentration to lower concentration. What about osmosis? What osmosis? Yes, solvent molecules across a selectively permeable membrane. All right. Diffusion moves solid, which is what is dissolved inside, and osmosis moves solvent. All right. Okay, so that's the two differences. And then mediated transport. This is wherever you have any molecule that assists, whether it be a protein or whatever, that assists in the movement. So what are some of the examples of mediated transport? Active, yes. Primary, this is primary. Secondary active, and we'll talk a little bit about secondary active and primary, because that's you kind of had a few questions wrong on the secondary active. All right. All that facilitated diffusion. Facilitated, it means something, a protein is facilitating the diffusion, moving that substance. Facilitated diffusion. All these are examples of mediated transport, as long as some protein is assisting in the movement. So what is vesicular transport? What's vesicular transport? Yeah, movement with a vesicle. What's that? Huh? It's okay. So yeah, movement with a vesicle. So what are the examples of vesicular transport? Yeah, all those. What else? The two major kind of movements of those sympod, and antipod are specific: exocytosis and endocytosis. All right, and then sympods and antipods are just moving them in and out. Sympod will do what? What will a sympod do? It moves two different molecules, though, in the same direction. Yeah, it moves two different molecules in the same direction. Yeah, there were little challenges because some of these, there are slight differences. An antipod, what does an antipod do? Yeah, it moves them in opposite directions. And then a unipod, it moves one molecule in one direction. Yeah, that's right. So the main forms of vesicular transport are exocytosis and endocytosis. What are the three different kinds of endocytosis? That's the first one. When you move solids, what's that? Yeah, phagocytosis is when you move what kind of substances? I just said it. Solids. Yeah, so you got phagocytosis. All right. And then what else? What other substances can you move other than solids? Fluids. And what's that called? Starts with a P? Again, starts with a P. Yeah. Pinocytosis, yeah. Pinocytosis. And then the other one, it's almost like mediated transport, but it forms a vesicle, so it's vesicular transport. What's that? No. It involves attachment to a of the substance that's being moved to a membrane surface receptor. Okay, so it's receptor mediated. Receptor mediated endocytosis. So whatever is being moved will attach to a cellular receptor, and then a vesicle forms around it, and that's how it's internalized. Okay, so those three types of endocytosis: solid, fluid, and here it attaches to a receptor. All right, so we'll kind of go through the definition of each of these. All right, and then move on to cellular organelles. All right, so this is just the movement of solute molecules in a solution from high concentration to low concentration. And is this an active or non-active transport? It's non-active. It's not. It doesn't use any energy. All right. Once there's equilibrium, there's equal movement of solids back and forth. Okay. So that's diffusion. And here, this is just the basic definition. The solution is a mixture of solvent and solute, right? And the solute is what's dissolved in there, and the solvent is the fluid in which that is dissolved. It could be, most of the time, the solvent is water, because everything is in an aqueous environment. All the reactions occur in an aqueous environment, so it's water. All right, and then concentration gradient is the concentration of the solid. Okay, and that's going to be important for osmosis, because when you have a high concentration of solute, you have less concentration of what in a solution? Solvent, yeah. And the solvent is water. So osmosis is movement of solvent, from high to low concentration. So a very concentrated solution will then have osmotic pressure to pull water towards it, because it's got less water molecules in it. It's got less solvent in it because it's got a whole bunch of solute in it. Okay? So that's where the concept of osmotic pressure will come through. And we will talk about that just now. All right? So it's just the distance, the difference in concentration divided by the distance between the two. Because then the longer the distance, the less the pressure, because then the substances have to move a longer distance, you know. But pretty much the higher the concentration, the higher the gradient. All right? So diffusion, I gotta switch on my little speakers. It's just a simple cartoon here.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Okay, very good. All right, so we'll move on to osmosis, which is similar to diffusion, except what things? We are moving solvent. What else is different from diffusion with osmosis? Yeah, it's got to be semi-permeable membrane, selectively permeable membrane. All right? So those are the two differences. All right? But it's the same concept, where we move from high concentration to low concentration still. So when you define osmosis, you've got to mention the selectively permeable membrane. You got to mention also that it's solvent molecules, it's water molecules that are moving still from high to low concentration. So you mention all those three things to get all the points. All right. So like I was saying earlier on, because water is a small polar molecule, it is able to pass through the lipid bilayer. But it does it not as fast as when you actually have an aquaporin, which is a protein channel that will span the membrane, and it will have a channel that will let only water through, because water is the right size of molecule to fit through the channel. All right? So that will occur rapidly. You find a lot of aquaporins in which system? Where might you find the aquaporins? What's that? Well, not so much as in which other system. The urinary system, yeah. Because the urinary system, what does it do? It's fluid balance in the body. So you need to retain a whole bunch of water, okay? And so forth. So those become very important, because all the water that is, filtered through from the blood has to come back, has to be reabsorbed. All right? So you have a whole bunch of aquaporins, and we'll talk about it in NP2 when we do the urinary system and so forth, in the kidneys. All right? So osmotic pressure, we're going to touch on it a little bit. It's the amount of pressure that a solvent, a solution with solids in it with a certain concentration will pull water towards it. So the more solid there is, the less water molecules there is, the higher the pull for water to come to that solution to dilute it. Okay? Because it's movement of solvent molecules from high to low concentration. So it will be the force required to prevent movement of water, because that's how you can measure it towards a solution. All right, through a selectively permeable membrane. Anything that is solid dissolved in it has a tendency to pull water towards it. And the extent to which it will pull water to it, which is dependent on how much solid there is called osmotic pressure. All right? And iso-osmotic solutions would have, iso means what? Same. So iso-osmotic will have the same, osmotic pressure, and there will be no net movement of water. Hyperosmotic would be higher concentration, right, than the reference solution or than what you're comparing it to. All right? So what is blood? Blood is fluid plus what inside of it? Yeah, red blood cells, cells all inside of it, white blood cells inside of it. Okay, so if blood is hyperosmotic, if it's got a lot of dissolved things in it, all right, compared to the fluid in those cells, what's going to happen to the fluid in the cells? It's going to move out, right, and all the water, and the cells will shrink, all right? And then if it's the opposite, where the solution, the fluid of blood, which is the plasma, right, it's got less concentration, right? It's hypo, which is the last one, hypoosmotic than the solution inside the cells, which means the cells will have a higher concentration fluid. So water is going to move into the cells. Eventually they will blow up. Okay? The reason I mentioned that is it's important to have the same isosmotic fluids in the plasma as well as the blood. Otherwise, you will have that problem where the cells will either shrink out or they will burst because they will take too much water. So it's important to control. So that's why you need all the homeostatic mechanisms to do that. And the pH has to be the same, about 7.4, which is neutral. All right? So that's the concept we just talked about here. Hypertonic solution, water will get in and eventually the cells will burst. And this is a good example. We use the red blood cells. All right? And then the shape of the red blood cells is not by accident. It is specific to go through the capillaries, to come out of the capillaries and get in. All right? So, so it will be able to squeeze through those little openings. So once you change the solution, if blood is not, kept at a constant concentration as the cellular concentration, the cytosol, in this case the fluid component, then this changes shape and it can affect a lot of things. And if it's even worse, it could burst or it could shrink all the way, and then it affects the function. So homeostasis is very important for all this. Homeostasis. Everything is to maintain homeostasis. All the cellular processes or to create molecules and proteins. All right? So that's an important concept. I hope we have time for this.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
All right. So if you were to explain how osmosis works, how would you put it? All right. One of the things that was said here is that anything that dissolves in water is itself polar, right? Okay. Did you see that the fluids, when you added the urea, were the same? Did you see that the same level, right? But still water passed from here to there. Why is that? What happens when you have a polar compound? Anything that dissolves in water will be polar. What happens? What did they say happens to reduce the amount of water? Because when we started, it was the same. I don't know if I'll be able to write here. I can't. But it was the same the same volume, which means there were the same water molecules in there. All right. What happens is that these water molecules will attach to the polar molecule. All right. Because they are both polar, so they will attach to each other. All right. Just like those hydrogen bonds that we talked about, those interactions of water molecules. So when you have the water molecules attaching to the urea, you have less free water molecules. Okay? So if I was to ask how osmosis works, you'd have to explain that. The water molecules will attach to the solute, which means there will be less free water molecules. And then the molecules of water from the other side will move in. All right? Because when we started, we had the same fluid, we had the same level of fluid. Okay? Do you understand that? Right? Okay. So it is because the water molecules will attach to the solute molecules, and therefore there will be less free molecules in there of water. So molecules will move such, and then the pressure that you're going to create by drawing, this is the osmotic pressure. Because now you have more water on this side and less on the other side. All right. Okay, so mediated transport, you have a process where there's a helper molecule or protein. Something is helping or assisting with the movement. All right? So that's where the mediated transport is. Something is assisting with the movement. It could, most of the time, it's a protein, or whether it's a channel protein or whatever. Okay, so it moves ions and molecules across the plasma membrane. All right. So something that assists. So because there is something that's carrying the protein or the substance that's being moved across, what happens? You're going to have competition, right? Because if you have the similar molecules, they're going to compete to attach to that carrier molecule. All right? And if they're going to attach, it's going to be selective. It doesn't just attach to anything, because the protein shape is going to be set, and it doesn't attach to anything. All right? And there's also going to be saturation, right? Because once all the protein molecules are attached, then you're not going to have any more to attach to. So the transport will have a finite rate of movement based on based on what? Okay, what will regulate the movement of this mediated transport? What determines the rate of the movement? Well, if you're on the right track, but what is doing the work? What is actually doing the movement here? The protein. So if you have more of the carrier proteins, you have a higher rate of movement. If you have less, it doesn't matter what the concentration is. Once they're all filled, then that's it. That's the rate it's gonna go. All right. So that's the concept of saturation. See, I want you to be in competition and selectiveness for mediated transport. All right. So be able to explain these characteristics of mediated transport. All right. All right. right, so we're going to talk about a few types of mediated transport. All right, so the channel proteins. Channel proteins are transmembrane proteins that have a channel that will fit a specific ion or a specific molecule. And what determines what moves through is the size of the channel pore. All right. If it's small, it will only be fitting small molecules. If it's bigger a little bit, it will fit a slightly bigger molecules. All right. So that's a channel protein. All right. And carrier proteins. You see the difference between carrier protein and channel protein? A carrier protein will attach to that substance, and then it changes shape, and then it will move the substance. Whereas a channel protein, the size of the channel just determines which size molecule enters through it and goes to the inside of the membrane. So can you describe the difference between a channel protein and a carrier protein? You can, right? Yeah, very good. And then, so this is the uniport, which we talked about, moves substances in the same, one substance in the same direction. A symport moves two or more ions or molecules in the same direction. All right. An antiport will move two or more ions or molecules in opposite directions. All right. All those are examples of mediated transport. All right. And then anything also that uses a certain protein, like the sodium-potassium pump, is also mediated transport. It uses energy from ATP to move substances. All right. So secondary active transport, you had a few problems with this in the LS module. Okay. Secondary active transport usually piggybacks on the energy of something that has been actively transported. Like, for instance, sodium is actively transported outside of the cell, right? Okay? And then, when it flows down its concentration gradient back into the cell, it will piggyback other molecules. For instance, glucose in the small intestine. That's how glucose is then absorbed. And it is moved against its concentration gradient. Okay? So secondary active transport is when you have a molecule that is actively transported and creates a gradient that when it is moving down its concentration gradient, it will piggyback on something else. Okay? So an example is sodium and glucose in the small intestine. That's secondary active transport. Okay? All right, so that's good. And this is an example of facilitated diffusion.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
All right. So the difference with active transport with this will be what? What will be the difference? What's the difference with active transport? We just said this kind of movement is moving from high concentration to low concentration, right? The only thing is that we are attaching it to a protein. Okay? So it's facilitated diffusion. So active transport does what? Yeah. Why? Because you're moving something against its concentration gradient. Like for instance, in the small intestine, you've eaten your meal and you've got a whole bunch of glucose and you've absorbed all the glucose from the cell. At some point, there will be a lot more glucose inside the cells than, in the small intestine, right? So you will have to move it against the concentration gradient to finish up all the glucose that has been digested in the small intestine, right? Okay, so, and then it will take energy when you're doing that. Whenever you're moving something from down its concentration gradient, you don't need any energy for that. Okay, so that's active transport, is to move things across substances against their concentration gradient. All right. So vesicular transport. All right. So this is you had a problem with this in the LearnSmart modules when it was asking about the movement of large molecules. All right. And some small ones too across the. We just talked about urea. We said urea is both large and polar, and it can't even go through the membrane. So how does it move across the membrane in terms of vesicles? It is just internalized by vesicles and is moved out in those pockets. Because the vesicle is membrane bound and it can attach and become part of the membrane itself, and it can, put stuff to the outside or put stuff to the inside. So the thing here is large particles. Why large particles? Because they cannot just go through the membrane. Okay, so you'd need a vesicle to kind of enclose it. That's the only way you can move them across the membrane, because they're too cumbersome to move through that fluid mosaic model arrangement of the membrane. Okay, so it's the large particles that we are talking about here that need a vesicle, like a little bag that will internalize and externalize. So when you internalize, it's endocytosis. All right. So movement of materials into the cell via a vesicle. And then phagocytosis, we talked about it. It's when you move solid, through the membrane. For instance, when white blood cells engulf, like macrophages, when they engulf bacteria, which is solid, that's why they're called phagocytes as well, because they use phagocytosis to engulf the bacteria, and then it is broken down in those vesicles. Those vesicles will unite with lysosomes and things like that, membrane-bound vesicles that contain, enzymes that will digest through or break down that bacteria. So pinocytosis, when you have fluids that are, internalized, the membrane will just engulf that part where there's concentration of these small molecules, and then it's taken up in forms of droplets or liquids, pinocytosis. All right? And then, of course, receptor-mediated endocytosis, and I think I'll show you a picture of this. The molecule that's being moved will attach to a receptor on the cell surface, and then the membrane will just wrap around it, and then it is internalized as a vesicle. All right? But the substance has to attach to a receptor first on the cell surface. All right? All right? And then, of course, exocytosis is a movement of substances outside. All right, and usually vesicles that come, say, from the Golgi body and so forth, carrying proteins and so forth, going to other cells. The vesicle just unites with the membrane; it becomes part of the membrane, and material comes out. All right, so exocytosis, and this just shows phagocytosis. This is a macrophage, engulfing a bacteria. All right, or when you break down red blood cells, worn out red blood cells in the liver, then macrophages do that, and then they're internalized and then mixed with a lysosome or a peroxisome if it's a bacteria, and then that will break it down. So this is just a summary of all the transport we talked about. From diffusion, secondary active transport, all the mediated transport, vesicular transport, all is and then it will give certain examples. Like, for instance, secondary active transport, where you have the sodium moving with the glucose when you absorb glucose in the small intestine. All right? And then endocytosis, exocytosis, endocytosis when macrophages engulf bacteria and so forth. All right. So this is just a summary of that. All right. And this is an example of endocytosis and exocytosis. All right.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Okay, well, it mentioned here low-density lipoproteins. They use that receptor-mediated transport. Why do you think low-density lipoproteins do that? What might be a reason for a low density? Low-density lipoproteins have more protein than they do lipid, right? So they will attach to something because they are more polar because of the protein content. They got more protein in it. So that could be a reason why you have them attached to a receptor first and then they're internalized. Low-density lipoproteins. All right? So all these movements will depend on the size or the chemical properties of what's being moved, right? Okay, good. So if you think of the transport mechanisms like that, then they are easier to remember because you know that this type of movement is going to move this type of molecule. You can associate that together, and it's easier to remember it than when you're just trying to say, okay, I'm going to remember endocytosis and then pino and phagocytosis and receptor-mediated or vesicular transport and things like that, just without associating reasons for the differences, because that's the only way to remember stuff without jamming them in your head. All right. So we move on to the cytoplasm. All right, and we talked about how the cytoplasm is the material that is enclosed by the membrane, okay, and it will have the fluid component, which is the cytosol, and it will have the cytoskeleton. So the cytosol is what you guys didn't know, that is the fluid inside of the cell, all right, and then it will have this cytoskeleton, which is the microtubules, actin filaments, all these long fibrous proteins that will place all the organelles in their positions or move vesicles when they move inside of the cell. These microtubules do that, all right, and half of them are organelles, all right, which are solid membrane bound organelles. Some of them are not membrane bound, like the ribosomes and the centrioles, and we'll talk a little bit. I think that's the last thing we'll talk about. There are certain membrane bound structures inside of the cell. They're sort of partial organelles in that when the cell produces something, it will package it. Let's say it produces a certain protein. All right. It could be melanin. Let's say melanocytes in the skin, they produce melanin, which is a protein, and then it will package them into these sort of temporary, bags or vesicles, membrane bound. So those are called cytoplasmic inclusions. Or it could be something that has been taken in via pinocytosis, for instance, and then that is a cytoplasmic inclusion. All right. So lipids, glycogen, like it is stored in the liver, right? When it converts glucose into glycogen, and then it is stored in these little bags. It's like storage, packing things in these little containers called vesicles, which are membrane bound. All right, and then because they are temporary, they're not like the mitochondria, which is always going to be there. The cytoplasmic inclusions could then be used when the glucose or glycogen is needed, then they're used up. When the melanocytes, melanin is needed, then it's used up. Okay, so they are kind of sort of temporary organelles. You could kind of call them temporary organelles. Okay, because they are membrane bound, but they get used up when they are needed. All right, so half cytosol, which is fluid with all these cytoskeleton and all these fibrous proteins that help to suspend the organelles inside of the cell and also move the vesicles around the cell. All right, so we're going to go through the organelles one by one, especially the important ones, and we'll talk about their functions. So these are basically specialized membrane bound structures inside of the cytoplasm, all right, that have a specific function, and we'll talk about that. So we all know about all these mitochondria, peroxisomes. Peroxisomes have certain type of oxidases in them, and lysosomes have just enzymes that will break, like worn out red blood cells, things like that. Peroxisomes are more used to break down microorganisms like bacteria and things like that. All right, and detoxify, substances as well. All right, so non-membranous is centrioles and ribosomes, and we'll talk about centrioles later on, which are located in the centrosome, very close to the nucleus. All right. All right, so the nucleus, of course, we talk about it specifically because of the genetic material that it contains, and it has two membranes binding it, demarcating it, and it's called the nuclear envelope. And then the nuclear envelope has pores on it through which the messenger RNA comes out during protein synthesis. All right. Okay, so you have DNA inside of it, and we said the DNA is not just occurring there. It is double-stranded, and it is folded up, and then it's attached to proteins called histones, and they form, once DNA is attached to the proteins, it's called chromatin. All right, and then chromosomes is kind of unfolded a little bit, and it forms certain strands like this. Usually they are attached right there in the centromere, and these are chromosomes. But when they are just folded up and attached to the proteins, it's called chromatin. All right, and these are chromatids, the two pairs in a chromosome, right? And this is only during cell division when they kind of sort of unfold, for obvious reasons, to protect. All right, and it's the centromere is what connects the two chromatids right here, and it's almost always not at the middle. It always connects them towards one side. The centromere never occurs in the center. That's just how it is. All right, and then so that's the nucleus. The nucleus has in it a dark staining regions called the nucleolus. Nucleoli plural, that forms ribosomal RNA, and it forms the ribosome subunits. Ribosome have got two subunits, something like that and something like that. All right, where the messenger RNA chain will kind of attach, all right? And when you get a transfer RNA coming in, it will kind of dock and it's held here. So this is tRNA over here, and it will carry the 3 codons which will attach to the carrying the protein. So ribosomes have 2 subunits, and all these are made in the nucleolus. All right. So it's the site of ribosome assembly, produces ribosomal nucleic acid, which is the nucleic acid, RNA that's attached to ribosomes in the cell. And this is the what makes the proteins that are used by the cells, because those proteins, as soon as they are made, they are released into the cytosol. All right. So ribosomes are non-membranous organelles that occur free or attached to the endoplasmic reticulum, right? All right. So ribosomes synthesize proteins that will be free because they are not membrane bound. The proteins are created and then they are already in the cytosol. So these are usually the proteins that the cell will use, right? And then if it's attached to the rough endoplasmic reticulum, then it will be protein that will be enclosed in a vesicle and then usually transported out. Okay, so you see the difference between the free ribosomes and the ribosomes that are attached to the membrane-bound rough endoplasmic reticulum, right? And then this produces proteins that will be secreted out. All right, so just know the difference between the free ribosomes and the proteins that are made by the rough endoplasmic reticulum. All right, so which is what we're going to talk about. So the main function of this is to create proteins. All right, create proteins, and it's long membranous sacs that have studded with ribosomes. All right, and then we have 2 of them. The ones that are studded with ribosomes are called rough endoplasmic reticulum, and the one that's not studded with ribosomes is called the smooth endoplasmic reticulum. All right. So the rough endoplasmic reticulum's main job is protein synthesis because of the ribosomes. When proteins are made, the messenger RNA attaches to the ribosome. All right. So major site of protein synthesis, makes enzymes and stuff like that. The smooth endoplasmic reticulum, on the other hand, I always associate smooth with something that's sleek, like a lipid. So the smooth endoplasmic reticulum is the site of lipid synthesis and carbohydrate synthesis. All right, but the smooth endoplasmic reticulum has different functions in different organs. Like in the liver, it creates glycogen and so forth. All right. Usually in the exam, sometimes I'll say, can you give me the function of the smooth endoplasmic reticulum in the liver? Or in the skeletal muscle and cardiac muscle, what does the smooth endoplasmic reticulum do? It stores the calcium that is used for muscle contraction. Because calcium attaches to a certain protein that is involved in muscle contraction. All right? And the smooth endoplasmic reticulum in the intestine, what's happened there? The intestine, that's where fats are absorbed. So fats are broken down in the digestive system, and when they are absorbed by the smooth endoplasmic reticulum, where they are reassembled again into fats, and then they are attached to proteins so that they form the chylomicrons to make them soluble, because everything is aqueous environment again. And all that occurs in the smooth endoplasmic reticulum in the small intestine. So you can see the different functions of the smooth endoplasmic reticulum in different organs, because I'm going to need you to be able to tell me these functions. The only function that doesn't have anything to do with lipid and carbohydrate synthesis is in the muscle, in the cardiac muscle, where it just stores the calcium. Okay, the rest of the other functions have to do with either the lipids or the carbohydrates. Okay, which is the major function of that. So, gotta know the liver, and then in the testes, production of the lipids, which are testosterone, right? Testosterone is a lipid, remember, because it's based on cholesterol. So again, in there, it's based on the true function of the smooth endoplasmic reticulum, which is fat, lipid synthesis, and carbohydrate synthesis, right? So the intestines. So know all these 4 organs and know the different functions of the smooth endoplasmic reticulum. So the Golgi apparatus is normally associated with the rough endoplasmic reticulum. What's the main function of the Golgi apparatus? It packages, collects all these proteins that have been made in the endoplasmic reticulum, and then they are packaged into vesicles, which are then via exocytosis moved outside of the cell. That's why the endoplasmic reticulum and all these membrane-bound organelles make proteins that are transported outside of the cell. All right? And also what it does, what else does it do besides packaging and so forth? It will fold up and into the tertiary structure. Remember, we talked about the primary structure, the secondary structure, the tertiary structure, and the quaternary structure, where they are associating with different proteins that are all folded up. So it will attach it to maybe lipids, or if it's going to be a glycoprotein, and then it will attach it to that, to carbohydrates, or whatever the case is, and to form the final product. All right? And then the good thing is that it is this membranous sacs that can form vesicles, and then it's externalized. All right? So another thing I ask about is all the functions of these membrane-bound organelles. Lysosomes, I said, these ones, they just contain enzymes, and they break down worn-out cells, for the most part, cellular components. All right? And they also digest bacteria and viruses and toxins, all right, and degrade non-functional organelles as well, so that new ones can be created. Break down glycogen, release thyroid hormone, and so forth. All right, and also any tissue that is no longer useful is broken down by lysosomes. All right, bone. At the stimulation by parathyroid hormone, you get the lysosomes breaking down bone to furnish calcium. All right. So lysosomes have more to do with just everyday cellular functions and cleaning up of cellular debris and things like that. So that's kind of like the overall function of the lysosomes. All right. Secretory lysosomes are found in the white blood cells. Why? Because when the white blood cells come across bacteria, they can secrete these enzymes so that they can start to digest out. So those are the basic functions of the lysosomes. Compare those to the peroxisomes. The peroxisomes are specialized in that they have oxidases that will take care of free radicals. Free radicals are reactive elements that have an unpaired electron, okay, such as superoxide, hydroxyl ions, hydroxyl radicals, and so forth. So peroxides have a lot to do with breaking down of the acids, fatty acids, amino acids, and hydrogen peroxide. Hydrogen peroxide is byproduct of those breakdown of fatty acids and amino acids. All right, so detoxify harmful toxic substances as well. All right, especially free radicals. Free radicals are very reactive molecules that have an unpaired electron, because electrons always occur in pairs, so it becomes very unstable, and so peroxisomes do that. All right, neutralize dangerous free radicals. This is the superoxide radical, superoxide. All right, and then hydroxyl radical has got a free electron. It's an OH with a free electron that's by itself, unpaired. All right, so major difference between lysosomes and peroxisomes is the detoxification of these radicals. All right, the mitochondria, this is the ATP, forms ATP via oxidative phosphorylation. All right, and it also has the same deal as the nuclear envelope, except the inside membrane forms what are called cristae. Why do you think it folds up like that? Why is it folding up like that, the second membrane in the mitochondria? Surface area again. That's why it folds up like that, to increase, because oxidative phosphorylation occurs on those membranes. All right, so that's the reason. is only other organelle that has its own DNA. All right. Mitochondrial DNA, so it can replicate by itself, I guess because of the specific function of producing energy. It doesn't need any direction from the nucleus to make its own proteins that it needs. All right. Okay. And the last bit is the centrioles. Centrioles, they are located in the centrosome, which is a very specialized area, very close to the nucleus. Why? Because the centrioles have all these microtubules that radiate from outside of it that are used to move DNA when you replicate. All right? So these are the microtubules. And each centriole has got triplets, 9 triplets of these microtubules, which are fibrous proteins made of tubulin. Okay? All right? And microtubules are formed there. All the microtubules that you find in the cytoskeleton, they are produced at the centrosome. All right? And the microtubules that move the DNA, and that's why it's during cell division, that's why it's located next to and they're usually the centrioles oriented at right angles to each other like that. All right? So these microtubules form the spindle fibers. The spindle fibers are the ones that separate the DNA during mitosis and meiosis. All right? So that's the centrosome, and then forms the basis of cilia and flagella. All right? I guess I never showed you an example of flagella. I drew it in, though, in that one. So that's it. Thank you, guys. Sorry, we went a little over, but the last bit is just the cilia and the flagella. And the problem you had in the exam, sorry, I went too fast on that, was just distinguishing between the microvilli and the cilia and the different functions. Surface area, movement of substances like mucus on top of the cell membrane. And then, again, this is just a summary table of all the organelles. And that's it. Thank you, guys. Everybody got a handout? Okay, good.